Myself, Dr. Deepika Sani from Lloyd Institute of Management and Technology. In the previous two parts of cholinelytic agents, we have covered the structure activity relationship of these anticholinergic drugs as well as the alkaloid solanaceous activity of solanaceous alkaloid as anticholinergic agents. So, in today's lecture, we will study about the synthetic cholinergic blocking agents. So, there are as we have studied earlier like in the case of solanaceous alkaloid or the synthetic cholinergic blocking agents, the process, the mechanism is same, that they will competitively, they will bind to the muscarinic receptor and will inhibit the cholinergic actions. So, in the case of synthetic cholinergic agent, the first example is tropicamide. So, it is very effective anticholinergic agents and specifically they are used for the ophthalmic any ophthalmic use for the eyes. For the eyes, like any inflammation in the eye, apart from that like in the cycloplasia that we have already mentioned, when the function of ciliary muscle is not proper, in those cases also, but it also produces short acting mydriasis. So, it is also used there, and to achieve the mydriasis, the concentration used is 0.5 to 1%. So, 0.5 to 1% of the tropicamide is used in the treatment of the to achieve the mydriasis. And in the case of acute iritis, that we have already discussed, in which there is inflammation in iris and iridocyclitis and also in the keratitis. So, these are the ophthalmic issues that can be resolved using tropicamide. There are some other examples which are used as synthetic anticholinergic agent. The example is a cyclopentolate. The name or arises from here, the cyclopentane group is there. So, cyclopentolate and clinidium bromide is also there, and the glycopyrrolate is also used as synthetic anticholinergic agent. And the next drug is dicyclomine, one of the important drug as anticholinergic agent, and it is used in the spasmolytic effect on various smooth muscles and specifically those who are associated with gastrointestinal tract. So, when there is spasm in the GI tract, the main drug that is used to cure that, for the spasmolytic effect on the smooth muscles, those who are associated with the GI tract. So, what will happen, this dicyclomine that will bind to the receptor, the cholinergic receptor present on smooth muscles, that those who are located in the GI tract, and will lead to its spasmolytic effects. And because of that, it is also useful in the dysmenorrhea and pylorospasm and also in the biliary dysfunctioning. So, in all these cases, dicyclomine is used because of its spasmolytic effect. This is the synthesis of dicyclomine. So, let us start with 1,5-dibromopentane. So, let us start here 1, 2, 3, 4 and 5. So, the bromine is present at position number 1 and position number 5. So, the 1,5 di means 2 and bromopentane, as there is no cyclic compound, we can write it in a straight manner also, like CH2 here Br, 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, and here is Br. This is CH2, 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 and so 1, 2, 3, 4, 5. This is 1,5-dibromopentane. and hence it is clearly this is a saturated compound and hence it is pentane. So, 5 carbon containing a saturated compound. So, 1,5-dibromopentane. Here we have taken benzyl cyanide in the presence of NaNH2. When we have we consider this, so it will lead to the cyclization of this compound using this CH2 here, and it is, so what was there, it was like CH2Br and here CH2Br. What will happen here? Here this was the benzene, that is CH2 and Cn. This CH2, this H2, it will extract this bromine and this bromine and complete this cycle. So, here we can say one cyclohexane, fine, one cyclohexane, it is connected with one benzene ring and one CN, it is connected with one benzene and one CN. So, hence I think it is very clear from here, there is here is the loss of 2HBr. This bromine atom, these two bromine atoms are actually removed by these two hydrogens, and hence there is formation of this intermediate. Further, by the by using acidic ethanol, this H3O positive and ethanol, it will get converted, this cyanide is being converted into COOO C2H5, the esteric form. Further, now what we have used, we have also used this moiety here. What is this is CH2, CH2, that is N, again C2H5 and C2H5, and it is oxy sodium ethoxy salt of diethylamine. So, this is ethoxy sodium salt of diethylamine. So, by the action of this, what will happen, this this ONA and this C2H5, this has been removed, C2H5ONA, this is being removed. Now, what will, this is COO and this will get combined with CH2 twice and NC2H5 and C2H5. So, it will get bind here. After the by reducing this benzene ring to the cyclohexane, it will get converted into dicycloamine. So, what happened here is, we have taken the hydrogen, we have passed through it to hydrogen using the platinum wire, and it will get converted into dicycloamine. So, the benzene ring, this unsaturation is being saturated by using the reduction method. So, this was the synthesis of dicyclomine. So, let us revise it once again. We have started with 1,5-dibromopentane, that we have seen this this is a saturated system. By the action of benzyl cyanide, what has happened, this H2 and these Br is being removed, 2HBr has been removed from this place, and that will lead to the cyclization of this long chain of 5 carbon atom, and hence there is a cyclohexane, that is there is an intermediate with a cyclohexane with a cyanide substitution and the phenyl substitution. Further, by the action of acid and ethanol, what will happen, it will, this cyanide, it has been converted by the oxidation method, it has been converted into acid and further the ester group. So, this COOH and further COOC2H5 by the action of ethanol. Further, this is by using uh, ethoxy sodium derivative of diethylamine. So, again there is loss of C2H5 and ONA from this side, and this whole group is attached to this COO. So, here we can say COO, CH2 twice, CH2 twice and C2H5 twice. Fine. So, after this we have reduced this because there is unsaturation in benzene, this phenyl group. So, what we have received, we have obtained the cyclohexyl group. So, this is the synthesis of dicycloamine. These are some other examples. This is methenathelin and this is biparidine, one of the drug used as anticholinergic agents, and this is isopropamide and this is ethopropazine. Further, Benztropine. Benztropine has anticholinergic as well as antihistaminic and local anesthetic activity. As we have already studied, some of the drug they will lead to the loss of the sensation of that particular area. So, these are also known as local anesthetic. Apart from that, they also inhibit the histaminic receptor in the same manner as that of the cholinergic one. So, they are also known as they are also known for their antihistaminic property. Its cholinergic effect, it make it applicable as anti-Parkinson agent. These agents are more useful in case of suppressing the symptoms of Parkinson disease. So, hence they are used as anti-Parkinson agents, and they are about a potent anticholinergic agent similar to that of the atropine. So, their activity is somehow potency is equivalent to the atropine. Next is orphenadrin. Orphenadrin, it is related to diphenhydramine when we talk about the structure, but antihistaminic activity is not as much as that of diphenhydramine. Diphenhydramine is a antihistaminic agent. So, it is the structurally it is somehow related to diphenhydramine, but the action is not. It does not possess a higher antihistaminic activity, rather it is more of anticholinergic action. So, it has a binding affinity more for the cholinergic receptor to antagonize the action of acetylcholine rather than to bind with the histamine receptor by to inhibit the action of histamine. So, hence it possesses lower antihistaminic activity and higher anticholinergic activity, but it also reduces the voluntary muscle spasm. So, it centrally when we talk about cerebral motor area, in the case of cerebral motor areas, it affect those areas and hence reduce voluntary muscle spasm. So, in the case of muscle voluntary muscle spasm, it reduce the activity of those voluntary muscle spasm. Next is procyclidine hydrochloride. This is a peripheral anticholinergic agent, as we have clearly seen that the nitrogen is quaternary ammonium. So, definitely it will not work through central nervous system and will not affect any will not possess any therapeutic or side effect through the central nervous system, but it is effective peripheral anticholinergic agent. Its ability to relieve voluntary muscle spasticity by its central action, and it is also used in the treatment of Parkinson syndrome, and apart from that it also reduces the muscle rigidity in the case of post-encephalitic and the arteriosclerotic and idiopathic type of diseases. So, in all these diseases, it is used to reduce the muscle rigidity. Like in some of the the example is arteriosclerotic. In this case, what will happen, there is a deposition of cholesterol in the cholesterol in the blood vessels, and what will happen, that will reduce the elasticity of those blood vessels and lead to the rigidity in those blood vessels. So, what will happen, by the use of procyclidine, for example, we can reduce those muscle rigidity, those muscles who are present in the blood vessel, that was responsible for the flexibility. But because of the diseased state, now the flexibility has been reduced and more of the rigidity is present in those vessels. So, this can be reduced by the action of procyclidine. This is the synthesis of procyclidine. So, we will start with cyclohexyl phenyl methanone. So, this is methanone, and on one side this is cyclohexyl and this is phenyl ring. So, after this we have used this bromo. We have taken this bromo as ethyl bromoacetate. This is CH2BrCOO and C2H5. So, this is ethyl bromoacetate by the action of the zinc. So, 1-phenyl ethylamine is also taken, and that will convert this C double bond O, this being the action will be done on this ketonic group, and hence this is 3-cyclohexyl. This is 1, 2, 3. So, at 3 position there is cyclohexyl, and at the 3 position there is hydroxy, and at the 3 position there is a phenyl, and this is 1, 2, 3. This is propionic acid. So, 3-cyclohexyl, 3-hydroxy, 3-phenyl, this is 1, 2 and 3, 3-phenyl propionic acid. So, by the action of this ethyl bromoacetate and with the 1-phenyl ethylamine in the presence of potassium hydroxide, we have synthesized 3-cyclohexyl, 3-hydroxy, 3-phenyl propionic acid. Further, B2H6 will play its role, the dimer of the boron hydride, and this will lead to the formation of, now what will we get, 1-cyclohexyl, 1-phenyl propane, 1,3-diol. So, here we started with 1, 2 and 3. So, at position number 1, now we have not started from this, because we have to number the longest chain, so that the substituent will get the least number. So, if we start from here, only one hydroxy will receive number 1 and the cyclohexyl phenyl and another hydroxy will receive number 3. Now, after starting this chain naming from here, numbering from here, so now at position 1 we have cyclohexyl group, we have phenyl group and 1, 2 and 3. Now, at position number 1 and position number 3 we have 2 alcoholic group. Hence, the name is 1-cyclohexyl, 1-phenyl propane. This is 1, 2, 3, 3 carbon chain. So, propane 1,3-diol. And further, with the help of carbon tetrachloride, triphenyl phosphine we have taken. This is phenyl, this is phosphorus, and we have 3 phenyl. So, this is triphenyl phosphine. By the action of this, we will get, now what will we get? We have replaced this hydroxy with the chlorine atom. So, now again 1, 2, 3, the same pattern, 1, 2 and 3, but now what will we have? We have 3 chloro, 1 cyclohexyl, 1 phenyl and propane 1 ol. So, this is 3 chloro, this is at third position we have chlorine, at first position we have cyclohexyl, we have phenyl, and this is the functional group, main functional group. So, propane 1 ol. By further, by the action of pyrrolidine, we have taken pyrrolidine here. So, this chlorine atom, this chlorine atom will be replaced by this pyrrolidine group. So, this is the synthesis of procyclidine hydrochloride. So, now what we have received, we have what we have obtained, this procyclidine hydrochloride. So, let us have a quick revision on the synthesis of procyclidine, and here we have started with the cyclohexyl phenyl methanol, and with the help of this ethyl bromoacetate and 1 phenyl ethylamine, it has been converted into 3 cyclohexyl, 3 hydroxy, 3 phenyl propionic acid. Further, with the help of boron hydride, we have converted this into 1 cyclohexyl, 1 phenyl propane, 1,3-diol. Further, we have triphenyl phosphine in the presence of carbon tetrachloride. Now, this hydroxy group is being replaced with the chloro group. So, now we have 3 chloro, 1 cyclohexyl, 1 phenyl propane, 1 ol. Further, with the help of pyrrolidine, we have synthesized procyclidine hydrochloride, the salt of procyclidine. So, this was all about all the anticholinergic drugs. In short, we can summarize all the anticholinergic agents, like we have studied about the solanaceous alkaloids, which was received from the plants of the family Solanaceae, and we have seen the levoforms are more active as compared to the dextroform. Further, when we talk about the synthetic derivatives, we have studied various derivatives. We have studied amino alcohol esters, amino alcohol ethers and simple amino ethers also, and there also we have studied that quaternary ammonium containing compounds, the derivatives will not cross the blood brain barrier, hence will not have any therapeutic effect that is associated with central nervous system. And apart from that, those who are non-quaternary ammonium derivative, that will cross the blood brain barrier and will also exhibit, apart from the ANS property, the cholinergic action, they will also possess the therapeutic action as well as side effects, those who are associated with central nervous system. And those who some of the drugs which are used because of their property, they are also used, have a limited action on the Parkinson disease as well as, and apart from that, basically all the anticholinergic agents, they are supposed to suppress the cholinergic action and are given before the surgery to suppress maximum of the secretions, we can say, like the salivary secretions or the GI tract secretions. So, all of these drugs have different type of medicinal uses. So, it was all about the anticholinergic drugs. Thank you so much.